Great. And I hope you also have actually Steven sir here waiting. Thank you very much, sir, for waiting for that we can start the session today. Before we
Most
start
welcome.
today's, yes. So, yeah, thanks for being there. I would like to obviously start with your introduction. First of all, it's an exciting session because we know that microfluidics is something which has been actually been discussed a lot recently as a very promising technology, not only for present, but also in future where we are anticipating a lot of changes into it and improvement. But at the same time, we have good tools right now available in the market as well. So today's session is application and validation of sperm preparation using Zymot. And who better than Dr. Steven Fleming, who is our today's speaker. He is not just a name in embryology. He's a pioneer. He's been working with CooperSurgicals as a director of embryology and also as an honorary associate at University of Sydney. And he is very well known that because he has created the first master's degree in assisted growth technology with Dr. Simon Fischel, another pioneer. And that was a program that has trained countless professionals globally now. We know he has been authoring and editing number of books, chapters, peer review articles, and his research spans for cryopreservation, endometrial physiology, endometriosis, oocyte maturation, and also sperm preparation techniques. So he has been pioneering in a lot of other worlds along with the methods of even improving sperm DNA fragmentation. So whether you're curious or how about the microfluidic sperm selection can enhance embryo development or can these techniques fit into our future of ART, I think we are in for a treat. So thank you very much, CooperSurgicals. Thank you very much, Dr. Steven Fleming, for being available with us today to enlighten our students, academy, embryologists, seniors, and colleagues for this another exciting session through Indira Fort Academy on application and validation of sperm preparation using Zymot. Please join me in giving warm welcome to Dr. Steven Fleming. Dr. Steven Fleming, the mic is all yours. Thank you very much.
Thank you very much for that kind introduction, Naval. I'll share my screen. So let's be this one. Again. There we go. So hopefully you can see that okay.
Yes, you can see the presentation.
Good. Perfect. And get that out of the way. It's better. Ah, good.
Yes.
Okay. So, yes, thank you again, Naval. And I'd like to thank the organizers of this webinar and Indira IVF for inviting me to speak to you today. And in this talk, I'll be presenting some data demonstrating the benefits of separating spermatozoa by their differential ability to swim along a microfluidic channel and swim up through a semi-permeable membrane within a sperm separation device known as Zymot. So I'll consider these different points. So what is the premise of the technique? What are its reported benefits? For which patients might Zymot be suitable? And in what circumstances does the protocol need to change? And I've added a little section based partly on a workshop that I conducted recently in Bangkok in Thailand that worked very nicely. So I'll also include a little section at the end on how you can validate your Zymot sperm preparation method. So the premise of the technique is that, of course, Zymot is a sperm separation device. It comes in two sizes, 850 microliter and 3 ml. And primarily it separates sperm based on their motility, partly, of course, by their morphology. However, notably, it is reported to be easier to use. It's more reproducible because of that ease of use, and gives better sperm quality post-prep than, for example, a standard swimmer. The microfluidic component of the Zymot sperm separation device lies between the inlet port that you can see there on the left, where the liquefied semen is injected into the lower chamber, and the semi-permeable polycarbonate separation membrane that you can see on the right, through which motile sperm swim into the upper chamber for collection. Now, interestingly, Reza Nosrati and his group down in Melbourne have shown that sperm velocity increases by 50% due to a shift from a three-dimensional to a two-dimensional pattern of movement when they're adjacent to surfaces, such as a microfluidic device. So one of the rationales for using microfluidics to separate sperm is that it more closely approximates the close contact between the female reproductive tract and sperm that are swimming up to find the oocyte. Now, given the generally accepted morphologically normal sperm head dimensions of 3 by 5 microns, why were 8 micron pores chosen for the polycarbonate separation membrane? Well, in this mechanistic study from Harvard Medical School, 3, 5, and 8 micron pore sizes were compared after 15, 30, and 45-minute sperm separations. And as can be seen, sperm motility was high under all variables. You can see that in the left histogram. But sperm recovery was actually maximal with 8 micron pores after 30 minutes. And you can see that in those the histogram on the right above the 8 micron section. Now, furthermore, using World Health Organization strict criteria and aniline blue staining, normal sperm morphology and nuclear maturity were similar in the unprocessed stock semen and sperm separated using a 5 micron filter, but significantly higher following sperm separation with an 8 micron filter. And again, you can see that in the two histograms on the left side of the slide. More importantly, perhaps, significantly lower levels of reactive oxygen species are generated, with the lowest levels of sperm DNA fragmentation being observed following sperm separation using an 8 micron filter. And again, you can see that in those two histograms on the right side of the slide. So in practice, depending on the size of the device, 850 microliters or 3 mils of semen is drawn up into a 1 mil or a 5 mil luer tip syringe and is slowly injected into the inlet port. And you can see the inlet port just above the Zymot symbol on each of the devices. And you have to take care not to introduce any bubbles beneath the sperm separation membrane. And then taking a fresh syringe, 750 microliters or 2.5 mils of sperm prep medium is drawn up, with 50 microliters or 100 microliters being used to prime the outlet port, which you can see in the top left-hand corner of each of these devices. And the remainder of the media is added dropwise from about 2 centimeters above the device to cover the entire membrane surface. And here you can see it animated. So that loaded Zymot sperm separation device is then put or placed into a 90 millimeter Petri dish with the lid put in place and is incubated in a humidified incubator at 37 degrees C for no longer than 30 minutes. The recovery rate is actually determined by the incubation duration. You can see the sperm swimming up into the upper chamber. Then up to 500 microliters, at least in this 850 microliter device, of the sperm prep is slowly aspirated from the outlet port and placed into a suitable culture tube or centrifuge tube until required for IUI or ICSI, or indeed washing prior to use for conventional IVF. Now, if it was the 3 ml device, typically you would recover 1 to 1.5 mils from that outlet port. So why 30 minutes, you might ask? Well, essentially, when using a polycarbonate separation membrane containing 8-micron pores, for example, the percentage of healthy sperm recovery is saturated after 30 minutes incubation within a 37 degrees C incubator, which suggests that the filter loses efficiency at longer incubation times, possibly due to sperm collecting near pores and blocking the passage of additional sperm, which is actually supported by the observation of an increased retrieval efficiency with diluted semen samples. Now, there are several benefits of sperm separation using Zymot, including increased lab efficiency, improved sperm quality, particularly lower levels of sperm DNA fragmentation, and these benefits are reflected in improved clinical outcomes. So compared to density gradient centrifugation in the upper panel, which was long considered the gold standard in sperm preparation, you can see that Zymot processing in the lower panel involves fewer processing steps, including avoidance of centrifugation, unless the sperm prep is to be used for conventional IVF, which I'll explain later. Therefore, the putative risks of increased sample handling and centrifugation are mitigated by the use of the Zymot sperm separation device. Now, many studies have shown lower sperm DNA fragmentation following sperm preparation with Zymot, mostly admittedly as abstracts, but backed up sometimes, however, in peer-reviewed articles, such as this one, using the Zymot multi-850 microliter sperm separation device. Fragmentation is associated with poor outcomes. That isn't a consistent finding. In fact, sperm DNA fragmentation is most reliably associated with increased risk of miscarriage. Indeed, there's no clear effect of sperm DNA fragmentation on fertilization rates following ICSI. Now, this study again demonstrates the very low level of sperm DNA fragmentation following sperm preparation using the Zymot multi-850 microliter sperm separation device. But interestingly, sperm DNA fragmentation levels following density gradient centrifugation are much lower here in this study than in the previous study. And this may be perhaps because of a less aggressive centrifugation at 300 G for 5 minutes, followed by 2 washes at 800 G for 5 minutes. So indeed, 2 washes may be better than 1 with density gradient centrifugation. Within the last 5 years, there's been an increasing number of reports linking sperm DNA fragmentation to both whole and segmental chromosomal aneuploidies in human embryos. And in some studies, these observations have been correlated with an increased risk of repeated implantation failure. Therefore, sperm preparation techniques that provide better quality sperm may help increase implantation rates. Also, within the last 5 years, there have been an increasing number of reports comparing euploidy rates in blastocysts that have been derived from oocytes fertilized with sperm prepared by either density gradient centrifugation in these blue histograms or using the Zymot sperm separation device in these magenta histograms. As can be seen, the percentage of euploid blastocysts is higher in all the studies represented by this histogram, with a significantly higher euploidy rate observed in 5 of these studies. So, which patients is Zymot potentially suitable for? So there are categories of sperm samples where separation using density gradient centrifugation may be preferable to using the Zymot sperm separation device. Nevertheless, Zymot sperm preparation may be adapted for use in all types of ART cycles, including those involving cryopreserved semen. Now, density gradient centrifugation is probably the preferable approach with non-ejaculated samples that have been, for example, surgically retrieved during PISA and TISA procedures, or with retrograde ejaculates retrieved from the bladder. It may also be preferable to use density gradient centrifugation if the semen samples are hyperviscous or have failed to liquefy. And it's been known that density gradient centrifugation has been shown to be particularly efficacious with semen samples from men known to be positive for viral infections such as HIV. And the same probably applies to semen samples found to display contamination with bacteria. Now, given that the volume of the endometrial cavity is approximately 450 microliters, the sperm prep volume of up to 500 microliters provided by the Zymot Multi, a 50 microliter sperm separation device, is very well suited to its application in IUI cycles. And indeed, one IUI centre down in Melbourne in Australia has been using it to very good effect. Of course, with IUI, you want to be using generally good semen samples. And if you are wishing to get an increased yield, then there's always the opportunity to use a 3 ml device rather than the 850 microliter device. Or you could resort to density gradient centrifugation. And if you use the 3 ml device, you're going to end up with 1 to 1.5 mils retrieved from the outlet port, as I told you previously. So in that situation, you'll want to concentrate that down closer to 450 microliters for use in IUI. In some cases of very severe oligozoospermia, so we think in terms of a total motile count of less than 1 million, Zymot sperm preparation is perfectly suited for use in ICSI cycles. In IVF cycles, a relatively good semen sample is of course required to obtain sufficient sperm for insemination, especially given that a wash of the semen of the sperm prep is required, as I shall explain later. Besides, if the total motile count of the semen sample is less than 20 million, ICSI is often advised. With frozen thawed sperm, you can apply the same guidance as in all other cases. So published data are currently limited for the Zymot sperm separation device with cryopreserved sperm. Nevertheless, current practice supports dilution of the thawed sample as you would normally with medium in a dropwise manner, and then loading the thawed sperm prep, the thawed semen into the lower chamber of the Zymot device as per usual. And fortunately, just this year, a prospective single center controlled study of 150 couples using fresh sibling donor oocytes and cryopreserved ejaculates was published. And in this study, at least 8 metaphase 2 donor oocytes were allocated to each couple, split into two arms. So one arm underwent ICSI with the controlled density gradient centrifugation process sample, and the other arm was processed with the Zymot sperm separation device. And in this study, they observed a significant increase in the percentage of total and top quality blastocysts post-Zymot processing, as you can see in this table. With intrauterine insemination, one study, a retrospective cohort study of 265 IUI patients where Zymot was used for 133 and density gradient centrifugation for 132. And even though the mean total motile count of the semen samples was lower in the Zymot group and the sperm prep concentration was significantly higher in the density gradient centrifugation group, nevertheless, the sperm prep total motile count was significantly higher in the Zymot group. And after controlling for confounding factors with multivariable regression, Zymot was found to have 3.5 times as much chance of producing an ongoing pregnancy. However, another study by Mutos et al. Found no difference between different techniques. So it's fair to say that for IUI, at least, we're awaiting better evidence. And some studies suggest that insemination of 5 million motile sperm gives optimal pregnancy rates. So in that case, a 3 mil Zymot device might be considered. So what about fertilization rates? Now, in most cases, Zymot prepared sperm have been used in ICSI cycles. And in those published studies, there appears to be no clear improvement in fertilization rates. So you can see the Quinn et al. Study 75.2 versus 79.4 for density gradient centrifugation. Zaha et al. Almost identical, 80.1 versus 79.8. Banti et al. 72 versus 68. And Godiwarala et al. Did find better fertilization, at least in one of their studies, where they saw 66.7 versus 56.1% fertilization, which was significantly better with Zymot. With IVF, there's a paucity of published data, but of course, better motility and lower sperm DNA fragmentation might impact the fertilization rate. And Adolphson et al. They published a study a year ago where they found no difference in sibling oocytes, so 81 versus 78% for density gradient centrifugation. And again, I'll go into more detail in that shortly. So with the publications, the retrospective longitudinal cohort study on the left of 88 patients with paired ICSI cycles, so were controlling for themselves for patient variability. Their initial cycle used density gradient centrifugation, and their subsequent cycle used Zymot sperm preparation, with a mean interval of 6 months between cycles. And that resulted in significantly higher fertilization rates following Zymot sperm preparation. However, when they did a later sibling oocyte study of 106 ICSI cycles, they still found a significantly higher fertilization rate with their Zymot sperm preps, but not quite the same extent as with the previous study. However, another retrospective longitudinal study of 53 patients, again serving as their own controls in consecutive cycles, this one found no significant difference in fertilization rates between density gradient centrifugation and the Zymot sperm separation device. And in this study, it was previously known as the Fertile Plus method, so don't be confused by that. But you can see in those left histograms, the fertilization rates, although they were slightly higher, with Zymot sperm preparation, they weren't significantly higher. But nevertheless, the Zymot sperm preparation in this study resulted in better blastocyst formation. And we know that blastocyst formation, to some extent, is dependent on the integrity or the quality of the sperm that's required for embryonic activation, genome activation. And they also found that the utilization and euploidy rates were higher, as might be expected. Euploidy rates were higher in younger women, less than 35 years old. So in what situations might the protocol need to change? So there are categories of sperm samples using density gradient centrifugation may be preferable, but essentially with Zymot for conventional IVF, then there is evidence that the protocol needs change, and this is now reflected in the updated instructions for use. So this study that was first presented at the 14th Biennial Alpha Conference in Lisbon was a preclinical validation of Zymot sperm preparation, followed by a clinical validation in just 11 cycles of conventional IVF. And what you can see in the upper table with that initial evaluation is that Zymot sperm preparation yielded a purer sperm selection with significantly increased progressive motility of the sperm, though the fertilization and utilization rates were similar. However, when they decided to extend this to all conventional IVF cases, they observed an unexpected reduction in fertilization and utilization rates, at least in the first 39 IVF cycles that they undertook. But this was normalized once a wash with a 5-minute centrifugation step at 300 G was introduced in concordance with that updated instructions for use. So you can see the fertilization rate went from 41.4% up to 65.1%. And the utilization rate increased accordingly following that washing step. So investigation of the value of a sperm prep wash is illustrated in this graph on the left, where sperm motility and progression is observed to decline at 37 degrees C post-processing with Zymot in the absence of a sperm wash, but is maintained following a sperm wash and centrifugation at 300 G for 5 minutes. And as can be seen in the histogram on the right, the percentage of sperm DNA fragmentation was observed to be unaffected by such a short and gentle sperm wash. In fact, if anything, it's even lower following that washing and centrifugation. So with conventional IVF, based on the available data, a wash step is strongly recommended. And as I just showed you, that internal testing shows that a 300 G 5-minute wash has no impact on sperm DNA fragmentation. And it also allows you the opportunity to move from your sperm washing medium to fertilization medium following that sperm wash. So it allows users at that step to adjust the final sperm concentration. So with clinical outcomes, the evidence for an impact of Zymot sperm preparation is rather conflicting. So a study was presented at ESHRE in 2023, reporting lower fertilization, but nevertheless better pregnancy rates following Zymot sperm preparation. However, as can be seen in this table, an earlier prospective randomized controlled trial of 386 unselected ICSI cycles Zymot with 192 and density gradient centrifugation with 194 revealed similar clinical and ongoing pregnancy rates. And in this study of density gradient centrifugation versus microfluidic sperm selection with Zymot, a total of 179 couples underwent 189 ICSI cycles, and in this case with donor oocytes. The mean implantation rate of the Zymot microfluidic sperm selection group was 57.7%, while that of the non-microfluidic separation group or density gradient centrifugation group was 42.5%, and that difference was highly significant. The mean live birth rate of the Zymot group was 42.9%, while that of the non-Zymot group was 30.3%. Again, that difference being significant. And what they noticed was that they observed a particularly higher live birth rate and implantation rate and blastocyst formation rate when the paternal age was equal to or over 45 years. So how can you tell whether you're, if you want to validate Zymot sperm preparation, whether you're getting good improvement of the sperm quality? Well, there are various ways in which you can perform preclinical validation of the quality of Zymot sperm prep, including, of course, routine semen analysis of progressive sperm motility. You can use computer-assisted semen analysis of curvilinear velocity, average path velocity, and straight line velocity. And you can use various assays of sperm DNA fragmentation, such as the sperm chromatin structure assay or the sperm chromatin dispersion assay. Or a very simple approach is to use the high hyaluronan binding assay. And I will explain the rationale for that. So we know that during epididymal transit, sperm undergo spermiogenesis, and mature sperm, having developed hyaluronan receptors via plasma lemma remodeling, are able to bind to hyaluronan within the extracellular matrix surrounding the oocyte. And indeed, a sperm's ability to bind to hyaluronan has been correlated with its cellular maturity, less chromosomal aneuploidy, less DNA fragmentation, increased chromatin integrity, and normal morphology, which is usually related to better fertilizing potential. And we also know that from this study, where acridin orange is used as a differential stain to measure DNA denaturation or to detect DNA damage in sperm. So in this photomicrograph of a pixie dish coated with hyaluronan, the selective power of that drop of hyaluronan is clear to see, with over 99% of the sperm within the circular drop of hyaluronan containing native intact double-stranded DNA, which stains green, compared to red-stained sperm containing damaged single-stranded DNA, which you can see outside of that drop of hyaluronan, but really very little, if any, inside the drop of hyaluronan. So based on those correlations between binding to hyaluronan and sperm DNA fragmentation, it gives you the opportunity to use the hyaluronan binding assay as a way of validating the quality of your sperm prep following Zymot sperm separation. So the sperm hyaluronan binding assay, which evaluates the proportion of sperm that express receptors for hyaluronan, can be easily combined with standard semen analysis, and that same principle can be used not only to assess the quality of a semen sample, but also to indirectly assess the quality of a sperm preparation method, such as density gradient centrifugation. So you can see in that bottom right graph where there's much higher binding to hyaluronan in the lower 90% fraction of the gradient compared to the upper 45% fraction of the gradient. And this, of course, can be applied to Zymot. So in this workshop that I conducted recently, there was moderate to good binding to hyaluronan in the semen sample prior to Zymot sperm separation. But following Zymot sperm preparation, the Zymot sperm prep was remarkable in that every single sperm that you could see was attached to the HbA assay, the hyaluronan on the slide, and rotating as you would expect, with the sperm tail being very motile and the sperm head being attached to the hyaluronan. So it's clearly a very nice way to demonstrate the quality of a sperm prep following Zymot sperm preparation, because of that correlation between hyaluronan binding and sperm DNA fragmentation. And we know that Zymot sperm preps tend to have very low levels of sperm DNA fragmentation. So what are the take-home messages from all of this? Well, of course, efficiency gains will be dependent upon the use and protocol used for density gradient centrifugation. But in terms of Zymot, there's no doubt that it's very easy to learn and use, and fewer steps are involved, at least for ICSI sperm preps. So time is saved while the separation is occurring in the incubator. There's less witnessing because there are fewer steps involved, and therefore there's also reduced risk of any errors. Because of the simplicity of using the device, it produces reproducible and consistent results between users. In fact, using density gradient centrifugation, I've always believed, is very much user dependent. It may seem simple, but to do a good sperm prep using that method does require considerable skill. And most importantly, perhaps, the Zymot sperm separation device has been consistently shown to reduce sperm DNA fragmentation. We're still waiting for definitive studies showing improved outcomes. So yes, there was that one randomized controlled trial that I showed you earlier, but we need more of those. And the links to aneuploidy and the reports of more euploid blastocysts following genomic analysis with PGTA are particularly encouraging. But elsewhere, we need more data for conventional IVF and for IUI situations. By far, the majority of publications have been on ICSI. But the bottom line is that Zymot does give you better sperm quality, and it's an easier sperm preparation method that allows you to get on with doing other things while the sperm are effectively separating themselves through the device. Some references with that. So I hope that was of some interest to you, and I'll be happy to handle any questions that you may wish to throw at me.
Yeah, thank you very much for that clarity about how Zymot works, the research analysis went behind, and what all things we yet need to verify with regards to further researches also. I think we have one question, but before we take on to that question, I would like to have just one thing to be known, that if anybody has to introduce Zymot to their lab, any precautions or anything first which they always need to keep in consideration before implementing it into the lab?
Well, like I said, the actual technique is incredibly easy, so I don't think there's any particular precautions there. There's certainly space on the device to attach the patient ID. Now, ideally, you would just do one of these separations at a time. but in reality, there may be several separations going on concurrently. So it's important to keep them in separate Petri dishes, to do your double witnessing of the labeling of the device, and try to keep the device stable and level when you're moving it around. Don't tilt it or anything. And most importantly, which again was evident from a Zymot sperm separation workshop on one occasion, where media had to be taken out of the fridge and wasn't allowed to warm up sufficiently. If you don't use media at room temperature, then the cold shock can affect the recovery rate of sperm following Zymot sperm preparation. So I think those are the obvious precautions to take. Otherwise, if there's any additional concerns, then you can always, for example, do a sperm wash, not just for conventional IVF. You could do that, I suppose, for your IUI cases and for your ICSI cases, if you were concerned. So, for example, if you're leaving the sperm prep for some time before using it, you might want to do a sperm wash to avoid that fall off in motility that can occur over time, admittedly over several hours. But those are the only things I can really think of that the obvious pitfalls, if you like.
Yeah, yeah. Right. So comparatively, it's pretty easier, and the general precautions has been taken currently are also has to be taken. Unfortunately, today we are not having Ritesh sir. Unfortunately, he is not available and he couldn't join. But we have along with us Priyata, who is also in charge of Indira Fertility Academy
Hi,
with
sir.
us.
Hello, Priyata. I can see there are some questions in the chat.
Yes, so I read the next one. So this is quite a long question. I think there are a combination of two questions here. The first one says, most papers compare sperm sorter devices with density gradient. But when we look at the direct Zymot method, the mechanism, although remains quite similar, just does not have the membrane filter. So may I know why studies do not commonly sperm sorter devices with direct Zymot?
Yeah. So, I mean, it's a good question. I guess the main reason is simply a pragmatic one, that most centers would tend to use density gradient centrifugation as their routine method of sperm preparation rather than swim up. I always used to combine the two, actually, do a density gradient centrifugation and then allow the sperm to swim up into the supernatant and use that sperm that swam up into the supernatant. But it's a good question because certainly in terms of the duration and the temperature that the separation is conducted at, it's far more similar between Zymot and swim up. And in fact, one of the concerns with swim up in the past is that the sperm are in close proximity to the semen sample during the swim up, and at 37 degrees C, you might get exacerbation of reactive oxygen species release, and therefore an increased risk of sperm DNA infection of the good quality sperm that are trying to swim away from the semen sample itself. So it's a good question, and I mean, it's interesting, thinking back to the mechanistic study with the prototype of the Zymot sperm separation device that they conducted at Harvard Medical School. One noticeable difference was that with the swim up, they had quite high levels of reactive oxygen species generation compared to the Zymot device at very low levels. So, I mean, there's some quite interesting things to learn just between those two methods, rather than just looking at density gradient centrifugation. So I think that's a reasonable question, but I think that's the reason why. Okay, so do you want me to answer that second bit about the DNA fragmentation index?
Yes, that would be great.
Yeah.
So the question is, for high PAFI cases where it's more than 50%, usually it's observed that the sperm quality is quite poor. So in this case, would Zymot be helpful?
Yeah, well, I think it depends what you're going to use that sperm prep for. I think if you're going to use it for ICSI, then maybe it doesn't matter if you don't, if you get low numbers of motile sperm, providing you have a sufficient number for the number of eggs that are mature and able to be injected. So, usually you're only looking for 10 to 20 sperm. But I think the important thing is that if those sperm are more likely to be the ones without DNA fragmentation, then it's worthwhile using any method that gives you the highest likelihood of that. In other words, the lowest percentage of sperm DNA fragmentation following preparation. And the available data that we can see from many studies is that the lowest rates of sperm DNA fragmentation are following Zymot sperm separation. So on that basis, I'd say hopefully it would be worthwhile using Zymot sperm separation rather than, for example, density gradient centrifugation, where you'll get a higher recovery, but you may not be so sure of selecting a sperm devoid of DNA fragmentation.
All right. So I think you have answered both the questions. Obviously, with regards to swim up, which was of an earlier question, the one of the most important thing which people also understand take care of is the contamination which direct semen can bring into the incubator and also into the lab, and then affects the culture. And that's the reason I think swim up was not being directly
I
used unless
see.
used outside the incubator.
Okay.
Yeah.
Yeah. But you don't have to introduce, you can have an incubator specifically for your sperm preps. It doesn't have to be
Right.
the same, it probably shouldn't be the same incubator that you're doing your embryo culture in. So I think that concern is easily avoided. And you can have, rigorous cleaning techniques for that kind of dedicated incubator, again, without risk of unwanted side effects on the quality of your culture environment. So yeah. Would you like me to
Right.
deal with these other questions running through?
Yeah, I think one of the first questions which also mentions is which material, which the sperm
Yeah.
separation layer, the membrane, which material is membrane made up of?
Yeah, it's just a polycarbonate. So as far as I'm aware, it's very similar, for example, to the polycarbonate membrane that's used in electrophoretic sperm separation, which again we published a study on that with John Aitken some time ago, and the polycarbonate membrane does not appear to be an issue with these sperm separation techniques, such as electrophoretic sperm separation and Zymot. So yeah, so simple polycarbonate membrane.
Right. Yes, Priyata.
So the next question asks about the cost effectiveness of this device in comparison with other sperm preparation methods.
Yeah, so that's a good question again, because yes, there are fewer steps involved, but you have to compare, I guess, the cost of the device itself versus the cost of density gradient material and centrifuge tubes. So with Zymot, you're going to be using more syringes than you are centrifugation tubes, I suppose. So, but you'll also be using a large Petri dish. So it's kind of a bit like swings and roundabouts. You in some areas you're using fewer materials, in other areas using materials that you wouldn't otherwise use, let's say with density gradient centrifugation. And I guess if you compared it with swim up, then obviously it's more expensive. But again, it's the cost effectiveness which is the question. So again, the effectiveness of the method maybe compensates some of the possible increased costs compared to some other methods.
Yeah, right. I think we need to also
Okay.
look into the consideration, the final outcome, which I believe is more important. At the end,
Yeah.
patient is spending a huge amount for the live birth, and when we compare that, if we know one technique can deliver better with regards to live birth rate, and I think that's what something has to be kept in mind.
Yeah, I mean, we often think of the cost to the clinic, but there's also the cost to the patient. So
True.
both emotionally and financially. So if you can, use a method that enables that couple to achieve a pregnancy sooner, then that's really, cost effective. So I think
Yeah.
that's a good point, Naval. Thanks for that. Yeah, very good point.
Yeah. Thank you. We have one question in Q&A section, and that mentions that does sperm selection using Zymot results in reduced reactive oxygen levels and enhanced mitochondrial membrane potential in spermatozoa from the men with globozoospermia compared to conventional preparation methods?
Um, Well, I can answer part of that question. So certainly it results in reduced production of reactive oxygen species. And, I don't know that we've got enough data really out there to answer the rest of that question. There are not many studies that I can think of that have looked at the mitochondrial membrane potential, for example, following Zymot sperm preparation. Maybe I need to search around a bit harder. But you because of the relationship between reactive oxygen species and the mitochondrial membrane potential, you might expect that if you're avoiding reactive oxygen species generation, then you will improve the mitochondrial membrane potential. So, but show me the data. I always say. So you'd want to see the evidence for that. But that's what you would hope to see.
Right. I think most of the clinics also have to keep it to their consideration that it has to be evidence-based at their own clinic. They need to look at it, they need to find it, pilot, and then they can implement it accordingly so that it helps them standardize it in a much better way. Because every clinic has some different way of working, different equipments, different manpowering, and the pilot at the respective clinic helps standardize the technique better.
Yeah, I think the Adolphson et al. Study that was published after that Alpha conference, that's a salutary tale, really. That initially they thought everything was fine. Maybe the issue didn't reveal itself because they were using good quality oocytes in the initial part of the study. But then when you open it up to a range, non-selected couples, that's where you need to be particularly observant of, whether the change in your approach is, achieving what you hope it will achieve without any untoward, undesirable results. So, yeah, I think you're quite right that everyone has to evaluate it in their own hands. But like I said, that there are people out there in various types of centers that are very pleased with the results they're getting. And so I always think that you should get in touch with the centers that are, getting good results to,
Right.
to get tips and tricks from them, and if there are any,
Yes.
and or just reassurance that that the standard approach will give should give you good results. I mean, some centers, for
True.
example, I know are not incubating for as long as 30 minutes, and they're very happy with that approach. It's quicker for them, obviously. But for example, if you're using this method for ICSI sperm preps, how many sperm do you need? So so I think there's all kinds of variations on this approach that you can learn and adopt in your own clinic to suit your processes better.
Yeah, right. Yes, Priyata.
So there's a question here which is kind of related to globosospermia. So this person says, if there's poor sperm morphology, but the acrosome is intact. So in such cases, can Zymot be helpful for selecting better sperm for ICSI?
Okay, so the sperm morphology is poor,
but
but the
acrosome is
acrosome
intact.
is intact and okay. So yes, essentially, because you'd hope that the acrosome is going to be intact in sperm, whether they're poor morphology. Well, certainly if they're good morphology, but to some extent if they're poor morphology. So you hope they aren't going to undergo a spontaneous acrosome reaction until they meet the oocyte. So on the other aspects of the Zymot sperm separation device, essentially, most importantly, perhaps producing those very low levels of or very low risk of sperm having fragmented DNA, then I think it's still going to be worthwhile, even in those situations where, even though the quality of the sperm is poor in terms of its morphology, that the acrosome is intact. So I think there are other benefits to be had, regardless of that type of sperm sample.
Yeah, but I think it also opens up another level of debate, which right now a lot of people are talking about is sperm morphology versus sperm motility as a mode of selection, and there's a lot of things now going around it. We'll keep it
Yeah.
out of it right now because we are lacking time. We are just 5 minutes above the schedule, but there are two more questions. Do you have time to quickly answer them?
Of course, I've got as much time as you want to run this for. So yes.
Thank you. So this it's related to the incubation time, both the
Yeah.
question. If there is a low motility, can the increasing incubation help? At the same time, if you increase the incubation, does it have an effect on the pore size of the membrane?
Yeah. So again, that mechanistic study where they looked at different incubation times from 15 to 30 to 45 minutes, they showed that it doesn't increase the recovery if you increase the incubation time from, for example, 30 to 45 minutes. In fact, it actually, strange enough, reduces the recovery. So, sperm maybe are just getting blocked in those situations. But yeah, I mean, I would hope that the polycarbonate membrane would remain intact for that period of time. I don't know of any evidence that increasing the incubation time beyond 30 minutes, sorry, in this case, beyond 45 minutes, whether that adversely impacts the membrane pores. So I just don't have the answer for that one. I don't know. But I think it's actually interesting that some centers are doing shorter incubations rather than longer incubations.
Yeah,
So again,
true.
we just need data really to see, in a bit more detail, how these different incubation periods of time impact the sperm quality. It may be that, I mean, we've got that initial study, but it's good to repeat these studies to see whether those findings are reproducible. Again, in different users' hands. But all we can go on is the evidence that we have published so far, that we've seen published.
So for the timing, we would take it as an ideal time would be 30 minutes or less, not more.
Yeah, so the instructions for use say 30 minutes. So 30 minutes should give you the maximal recovery with maximal motility as well. But if you're not so concerned about recovery, then as I said, some users are reporting doing shorter incubations. So you could argue that a reduced incubation time shouldn't have an adverse effect on the sperm quality, at least. But you won't get as high a recovery, of course.
That's true. I think we're done with the questions, sir.
Yes.
Wonderful. Well, I'm very pleased to see such a big turnout. It's nice to have so many members of your academy and your different IVF centers turning up to tune in. So that's very nice and rewarding.
Yes, thank you so much. Thank you for your time. I think, yes, you clearly mentioned a lot of stuff and some of the questions which are there in the chat box. I think the answers were in the presentation, as you rightly said. But yeah, the query keeps bubbling up into the mind of an embryologist, though they have evidence, but they want more clarification, and you clarified all of that. So thank you. Thank you very much for having your time being spared for the academic students. At the same time, this sessions remain open for others also who want to join. So this increases the reach and whosoever wants to join in. So that's something which Indira Fertility Academy has been doing, trying to educate the people through not only from Indira, but also outside. And it helps sharing the knowledge. So thank you for being available for such an innovative, kind of a session. And we hope that we come across with some more other sessions with you, some more interesting talks. And I hope definitely for all rest of our audiences, the next month with another speaker, with another topic, we'll come back, and we hope that you like it and you keep attending the sessions as you always do with good attendance.
Yeah, my pleasure. So I'm very happy to come back at some future time to discuss a different subject.
Yes. Okay, so thank you very much, everyone. Thanks, Priyata. Thank you, Stephen. So thanks, CooperSurgical. And till the next month with the next topic and next speaker, have a wonderful evening and wonderful month. We'll come back with another sessions. Thank you so much. Goodbye for now.
Look forward to it.
Thank you, sirs.
Bye-bye.
Thank you, everyone. Bye-bye.